Hello everyone, and welcome to today's Labtools webinar. My name is Nathan Nee, Associate Science Editor for The Scientist, and I will be moderating our discussion today. Today, our speakers, Kristen Butcher and Cuoto-Rodriguez, will discuss the development of a nucleic acid hybridization capture-based assay to detect and identify novel SARS-CoV-2 variants. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and the speakers will address these during the Q&A session following the presentations. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. We will try to address as many of these questions as we can during our Q&A session. This webinar will be archived on the Scientist's website, and we will send you the link for the on-demand version via email within a couple of days. Please note that you will not be able to download the presentation slides. I'd like to take this opportunity to thank our webinar sponsor. Twist Bioscience is a leading and rapidly growing synthetic biology and genomics company that has developed a disruptive DNA synthesis platform to industrialize the engineering of biology. The core of the platform is a proprietary technology that pioneers a new method of manufacturing synthetic DNA by writing DNA on a silicon chip. Twist is leveraging its unique technology to manufacture a broad range of synthetic DNA-based products, including synthetic genes, tools for next-generation sequencing preparation, and antibody libraries for drug discovery and development. Twist is also pursuing longer-term opportunities in digital data storage in DNA and biologics drug discovery. Twist makes products for use across many industries, including healthcare, industrial chemicals, agriculture, and academic research. Our sponsor has provided us with some helpful resources related to today's webinar, and we have posted these in our handout section located on the right side of your screen. You can access and download these documents at any time during the webinar. With that, let me introduce our first speaker. Kristen Butcher joined the Twist Bioscience Research and Development team in 2015. Prior to this, she focused on developmental evolution and next-generation sequencing in the academic and government sectors. Butcher is currently a senior scientist at Twist and plays a leading role in developing the hybrid capture product line, helping to advance this technology for new and novel applications. Butcher earned a bachelor's degree in neuroscience from the University of California, Santa Cruz, and a master's degree in environmental management at the University of San Francisco. Let's just make sure everything is up and running and everything looks good. So, Kristen, please take it away.
Thank you so much for the introduction, Nathan. My name is Kristen. The Twist Bioscience and Biotia teams have spent the last year designing the highly sophisticated SARS-CoV-2 NGS assay technology and software to be extremely sensitive and useful for the detection, characterization, and identification of the SARS-CoV-2 virus. So we are super excited to be talking to everyone about it today. Here is our presentation on achieving highly sensitive viral detection with the SARS-CoV-2 NGS assay. So before we start, I'd like to take a second to explain what Twist Bioscience does. I know Nathan briefly went over it, but we are a synthetic biology company that prints and manufactures DNA sequences for various applications. Most of what I'm going to talk about today will be in our NGS product line. And so Twist offers DNA custom panels specific to your region of interest to be used in hybrid capture. We offer these as double-stranded DNA for the highest capture efficiency and exceptional uniformity. We also sell library preparation, target enrichment, and NGS reagents. I plan to define some of these terms for some of you who aren't familiar, but I want to mention that before I start defining these terms, I will be using the words probes and panel interchangeably throughout this talk. For those of you who are familiar with this technology, like my colleague Richard here on the right, we are super excited to talk about it, so let's get into it. A brief outline as to what Mara and I are going to cover today. I'm going to talk about the differences in viral detection assays, give a quick overview of what hybrid capture is, and then focus on the benefits of hybrid capture for detecting, identifying, and characterizing viral threats. Mara is going to take it over from there and show the end-to-end workflow of the SARS-CoV-2 NGS assay, as well as an overview of the COVID-Dx software and the data you get from it. She's then going to cover some SARS-CoV-2 variants of concerns we've been able to detect and identify using the assay, as well as compare our system with Amplicon-based assays. And so when determining the right type of test to use, it's important to understand the properties of the test. It's also critical to understand how the virus itself biologically maintains its infection. And so there are currently over 230 SARS-CoV-2 FDA authorized assays to choose from at the moment. And so this can be a difficult decision at times. The figure on the left here shows how the SARS-CoV-2 virus concentration changes over time. So early on in the infection, the concentration of the viral particles or viral load is higher. But as time passes, the virus becomes lower in viral load. So the two horizontal lines here in the figure, they represent two kinds of test sensitivities. The higher line represents tests that have low analytic sensitivity and are less likely to show up positive when the infection is in the beginning or ending stages. And in contrast, the lower line here, this represents high analytic sensitivity tests that are more likely to show up positive throughout almost the entire infection. And so therefore, the type of IVD test used can really impact the results of a test. And so this is due to the sensitivity of individual assays and fluctuating viral load throughout the infection. Not only does sensitivity play a role in what tests you should use, but the virus itself does as well. And so what you see here is a snapshot of emerging SARS-CoV-2 variants using our twist controls. At the top of the figure is the wild-type SARS-CoV-2 virus that started the COVID-19 pandemic. The horizontal gray areas below symbolize the different variants that came after the wild-type variant. The red, green, blue, and orange lines within the gray area represent different kinds of mutations found in emerging variants. Some variants emerge 1 month after the original wild-type virus, while others emerge many months after. And so you can distinctly see different mutations in each variant of concern. And unfortunately, the evolution of the SARS-CoV-2 virus does not have a pause button. It's not going to stop anytime soon. And so because of these mutations, some variants have been found to evade certain kinds of tests. More recently, we've seen this in PCR-based detection assays when trying to detect for the B117 SARS-CoV-2 variant of concern. Another characteristic some organizations may be interested in is related to multiple pathogens being tested for in one assay. So for example, SARS-CoV-2, RSV, and H1N1 viruses often show common symptoms, and it sometimes may be unclear to a physician what is causing the symptom. This kind of test may be suitable for some organizations. And finally, all assays are different in the type of data output and the hands-on time required. So this is something else you'd want to consider. There are three main methods for detecting viral infections. And so the table on the right here, it explains this well. So broken out into columns, there are three types of tests. Molecular assays detect for the presence of the viral nucleic acid sequences. And so these assays are typically highly sensitive and are able to detect if the patient is currently infected with the virus. Antigen tests, in the middle column, these are used to detect for the protein that the virus generates. And so these are typically lower in sensitivity and less accurate, but they take very little time, sometimes only 5 to 15 minutes, depending on the test. Serological tests are used to detect for the antibodies that the patient's body would typically release if infected with the virus. So these are also lower in sensitivity. And a positive test doesn't necessarily mean that the patient is currently infected. This can mean that the infection happened months ago. And so the type of assay we're going to focus on here are molecular assays, which is highlighted in teal, excuse me. Among all of the molecular assays used to detect for the SARS-CoV-2 virus, the two most commonly used are PCR-based and sequencing-based assays. The figure at the top of the slide here is an example of how a typical PCR-based assay would work. Primers targeting specific sequences of the SARS-CoV-2 virus are generated and added into a PCR reaction with the patient's sample, and any present virus would be amplified and detected. These kinds of tests, which use real-time PCR or digital PCR, are extremely focused and sensitive, but are only limited to detecting the virus. These are extremely short partial pieces of the viral genome that are amplified out. And so sequencing-based assays, on the other hand, these amplify out the entire viral genome and use next-generation sequencing, or what we like to call NGS. This technology not only detects, but it also identifies the virus. And Amplicon sequencing uses a large number of PCR primers to generate amplicon regions throughout the viral genome, while hybrid capture, otherwise known as targeted sequencing, uses nucleic acid probes to enrich for the viral genome from complex sample inputs. And so both of these methods are very comprehensive as well as sensitive and can also be used to identify the virus in most cases. The SARS-CoV-2 NGS assay is hybrid capture based, which I will show you in detail in the coming slides. And so we generated this table here to show the pros and cons of both PCR and sequencing-based assays. They can detect the virus in high sensitivity depending on the assay. Hands-on time is a bit longer for the sequencing-based assays compared to the PCR-based assays. But like I mentioned in the previous slide, sequencing-based assays can identify the virus rather than just detect it. Sequencing-based assays can be used to characterize evolving viruses and give researchers the accessibility to look for mutations of interest. They can also be used for tracking and surveillance purposes. And by being able to survey the virus and determine which viruses are prevalent in a community, we can better understand the effectiveness of public health measures and how well they work on specific variants. Now, why is sequencing a whole viral genome important? Understanding viral evolution and being able to survey how SARS-CoV-2 is spreading is extremely important. Going back to this figure that I've already showed you, I want to point out a couple more things here. The 4 bottom gray areas represent 4 major recent variants of concern: the B117, B1351, P1, and B1617 variants. You'll notice in these variants of concern, large amounts of mutations are accumulating in the spike protein region represented in orange at the bottom of the figure. This is what the majority of the vaccines use to establish immunity in humans, and therefore it's extremely important that we know how this region is mutating and evolving over time. Both amplicon and hybrid capture are 2 whole virus sequencing approaches. And so before I get into what exactly hybrid capture is, I want to give a quick overview of basic next-generation sequencing workflow for those of you who are not familiar with it. There's several types of NGS platforms. So here, I will outline NGS using an Illumina platform. Typically, you start with sample preparation that would include collecting your specimen via something like saliva or a nasopharyngeal sample. And that's actually why I have a little swab here. From there, you extract and isolate your RNA and then use reverse transcription enzymes to change the RNA to DNA. Then comes library preparation, which includes putting your DNA specimen into the reaction, fragmenting it into smaller pieces, and adding barcoded adapters onto each DNA fragment. And so this ends up being called the library. And each library includes a unique barcode adapter specific to that library so that once it's sequenced, the data can be pulled out. The pool of libraries are put onto an Illumina sequencer and the adapters bind onto the sequencing flow cell. Each base is then fluorescently labeled and called on the sequencing platform depending on the color that it fluoresces. The heat map on the right here, this is data that you get post-sequencing showing how well your libraries bind to the flow cell. And so once the sequencing is complete, the analysis starts, which includes file conversion, demultiplexing of the barcodes, and alignment to the target sequence. So now that I've outlined next-generation sequencing for you, you're probably wondering, why can't you get the sequencing data you need for viral genome using this workflow? So that's not really possible using the previous workflow. Adding an entire library onto a sequencing platform is often referred to whole genome sequencing. And so whole genome sequencing is unbiased, meaning whatever is in the original library will be sequenced. And for viral detection, we are specifically interested in sequencing small concentrations of the viral genome found in the sample, and we don't care about the human background. We need our sequencing to be more focused and targeted so that it's cost effective and we get the data that we need. And so read density, which is defined in this figure, is the percent coverage across the entire viral genome. We rely on this metric for evaluating how well we are able to identify the virus. Whole genome sequencing gives very poor coverage across the viral genome because most data generated is human background material, while hybrid capture gives great coverage across the viral genome. I will introduce this technology in the coming slides. And so the hybrid capture workflow looks something like this, with the most essential change being adding a target enrichment step right in the middle of the NGS workflow. This is used to enrich for areas using a custom panel made to target your region of interest. The main reasoning behind adding a target enrichment step to an NGS workflow is to amplify out the regions of interest so that you can increase your sequencing coverage and lower sequencing costs. And so think of this as a needle in a haystack metaphor. The needle represents your region of interest or target. And in the case of the SARS-CoV-2 NGS assay, this would be the SARS-CoV-2 virus. The haystack is the remaining excess library that you don't want, which would be the human background sequences. By targeting the material you want, you're able to sequence more of your region of interest as opposed to everything else in your sample. And so getting back into the details here, this figure summarizes the hybrid capture workflow. The pre-hybridization step includes generating a multiplexing pool by adding your bar-coded libraries together. The custom panel that I was referring to in the previous slides is added to the library pool. This panel is biotin bound, which is a detail I will revert to later. Both blockers, the universal blocker that's specific to adapter sequences and the blocker solution that's specific to known repetitive off-target regions, is also added into the pool. Once all of this DNA is concentrated, hybridization solution is added and the pool is left in a heated incubation period of 2 hours so that the panel has time to bind onto complementary DNA found in the libraries. In the binding step, magnetic beads that contain a streptavidin molecule are added to the hybridization reaction. Biotin that's bound to the probe is attracted to these streptavidin beads, causing the beads to capture the library containing the target of interest. And so wash steps are then added to get rid of any remaining off-target sequences, and the leftover target libraries are then amplified and cleaned up so that a majority of the sequences that you put on the sequencing platform are libraries containing the target region. And in the case of the SARS-CoV-2 NGS assay, the gDNA libraries going into the workflow represent your sample that contain extremely small amounts of SARS-CoV-2 virus with large amounts of human background material. The target-enriched libraries exiting the workflow represent your samples that have been enriched and contain a much higher percentage of SARS-CoV-2 sequences so that a majority of what your sequencing data is useful to you. And so uniform capture starts with uniform DNA synthesis. And so what I mean by this is getting uniform capture really starts with how well your target probes are made and amplified in relation to the GC content. And so the figure at the top represents a panel that was generated with the appropriate amount of probes, depending on the GC content throughout the entire region of interest. When this is not done correctly, you will end up with a probe that gives metrics similar to the graph you see on the left here. You'll notice by looking at the red line in the figure that there's more read counts containing higher GC content represented in the sequencing data. This ends up giving you non-uniform capture and uneven distribution in reads, some being over-sequenced and others being under-sequenced. At Twist, we use an optimized method for amplifying our probes so that we can get uniform probes, demonstrating an even distribution in sequencing, which can be seen in the figure to the right, represented by the red line. And so with uniform capture comes uniform coverage. When probes are amplified uniformly and sequencing reads are evenly distributed, there is no under or over sequenced areas of your target. More uniform coverage isn't just solved by probe design, but workflow optimization as well. You can see on the figure on the left that this causes our targets to be sequenced uniformly. The figure on the right shows the same data in a different way, comparing our output data to competitor data. We see more bases being read for a longer amount of time on the y-axis as depth of coverage increases on the x-axis compared to competitor data. A greater depth of coverage can increase confidence in final results. So ultimately, more uniform coverage means that there is improved coverage for more sequences overall, leaving less reads overrepresented, leading to lower sequencing costs. And how does this save in sequencing cost? By having the same percentage of sequencing for every target, regardless of GC content, prevents you from having to resequence or leave room for extra sequencing as a safeguard. And so when the COVID-19 pandemic started in late 2019, Twist responded by applying our hybrid capture technology to viral detection. We knew that we could do this because of previous product launches, for example, our Panviral V1 panel. We also knew that this would be crucial specifically for characterizing and identifying future variants that may come up. We pretty much already had our workflow already planned out for us. So we started by designing a panel specific to the SARS-CoV-2 virus. So this panel that we designed, called the Twist SARS-CoV-2 Research Panel, targets the entire SARS-CoV-2 virus, including the poly-A tail. It is designed to the original Wuhan 1 reference strain, but it is tolerant to mutations and variants, which I'm going to talk more about in the coming slides. It's a total of 994 unique probes. It was released as a standalone product in April 2020. And in November 2020, the RUO product with Biotia software formed the basis for the SARS-CoV-2 NGS assay, which was authorized for emergency authorized use by the FDA in March 2021. And so when Twist started developing the SARS-CoV-2 NGS assay, we validated the panel by using different concentrations of the SARS-CoV-2 synthetic control in human background to simulate a real sample. So what you see in the figure to the left is viral control spiked in at different amounts. We ran these samples through hybrid capture using the SARS-CoV-2 panel and asked the question, are we able to detect the virus post-enrichment? And we could detect it. The figure on the left shows this data at 1x depth of coverage. The table on the right emphasizes data even more, showing that as the virus copy number in the sample decreases, the fold enrichment increases, which means we are specifically able to pull down the viral sequence from non-specific sequences. You can see how dramatically the viral fraction between pre and post capture changes in the table to the right. It's like the needle in the haystack metaphor I mentioned before. When there is less virus in the sample, we want more amplification to make it more abundant, and therefore increase fold enrichment, which is what we see here. We can enrich for extremely low concentrations of virus. Thinking back to the graph in the beginning of this presentation, the SARS-CoV-2 NGS assay is very much a highly sensitive analytic assay that can detect the SARS-CoV-2 virus pre and post the infection stage. And so now that we know our panel works to enrich for the exact same sequence, how does it work when variants are added to the system? Can the panel still enrich for the variant viral sequences? This figure shows the effective random and continuous mismatches in a panel used in hybrid capture. We tested the effectiveness of mismatches in probe binding by adding continuous and random mismatches into a panel of 120 base pair probes. In a real virus, random mutations are things such as point mutations, while continuous mutations are sequencing insertions or deletions. We then ran hybrid capture using these panels. The results here show that we get 50% efficiency in hybrid capture using approximately 33% of continuous mismatches or 8% of random mismatches compared to the original sequence, suggesting that hybrid capture probes are robust even when there are mutations present in the sequence. So tying this back to both PCR-based assays and Amplicon-based assays, both types of assays use very short primers that are approximately 20 to 30 base pairs long. One mutation in a primer of that size can account for 5% variation. Effectiveness in mismatches in shorter primers would have much more of an impact in not being able to accurately bind to the complementary sequence and miss important data collection. And so next, we wanted to see if our panels would work on novel viral sequences. And so you've seen this figure a couple times before now, but I wanted to bring it up again. Can we detect for a virus that has the kinds of novel mutations that SARS-CoV-2 contains today? And so what we did here was design synthetic controls for the H1N1 virus with random mutations in the H8 region to simulate a real set of viral mutations. You can see on the x-axis what percentage in variation from the wild type that we designed for. We used a panel that enriched for the wild type of the H1N1 virus of years past. As the variation in each control gets greater, we see capture efficiency go down, which is to be expected. That's shown on the figure on the left. Comparably to the previous experiment we did, we get sequences with approximately 4% variation at 50% capture efficiency. This change is most likely due to the hybrid capture chemistry and target sequence differences between the two panels. However, you can see in the figure to the right, we were still able to get significant coverage along the entire virus. This means that despite the novel mutations found in the sequence, we were still able to get enrichment and enough coverage to be able to get 1x coverage across the full viral genome with up to 10% variation and at least 50% of the viral genome coverage at 1x with up to 20% variation. So our hybrid capture system and panels are designed to be able to handle these mismatches and enrich for novel viruses containing mutations that have never been seen before. And this is critical to identifying the SARS-CoV-2 variants of concern that we are discovering regularly. And so to recap here, I've talked about the different kinds of SARS-CoV-2 detection assays you could use and why sensitivity is important when choosing the type of assay that is used. I've also explained what hybrid capture is and how it's beneficial to any NGS workflow. And finally, I've demonstrated why hybrid capture technology in the SARS-CoV-2 NGS assay is an integral product that allows for not only the detection, but also the identification, characterization, and surveillance of the SARS-CoV-2 virus. If you'd like to find out more information on our tools and product lines, please visit us at our website at twistbioscience.com. And this is where I will pass it over to Nathan for the next portion of the talk.
Thank you, Kristen, for that presentation. As a reminder to our audience, you may submit your questions to our speakers using the Q&A tab at any time. We'll have a chance to address them after our next speaker. As Kristen mentioned, our next speaker is Mara Cuoto-Rodriguez. She has over 15 years of experience in microbiome research, fungal genetics, and bacterial diversity. As a senior staff associate at Columbia University, she conducted research on a diverse range of human microbiome projects employing next-generation sequencing techniques. This research provided insight into the role of the microbiome in health and disease, including stillbirth, HIV, bacterial vaginosis, colorectal cancer, and maternal antibiotic exposure during pregnancy. Cuoto-Rodriguez is currently the Director of Research and Development at Biotia, a New York City-based startup focusing on clinical diagnostics and surveillance by leveraging genomics and AI for precision infectious disease insights. Cuoto-Rodriguez received a master's degree in fungal genetics from the University of Georgia and a master's degree in microbiology and a bachelor's degree in biology from the University of Puerto Rico, Mayagüez. Let us once again make sure all of your slides are up and running and everything looks good. So please take it away.
Thank you, Nathan, for the introduction, and Kristen for giving such great insights of Twist technology. So today I will be complementing Kristen's talk by covering our end-to-end solution that we have co-developed with Twist to detect and survey our SARS-CoV-2. In particular, I will be focusing on the clinical validation of the assay, the software component and data that comes along with our clinical reports, and some more data on some projects that we have run in our lab. So just as a matter of a recap on some of these things that Kristen has been talking about, we know that SARS-CoV-2 is evolving. This is a natural trait of viruses as they replicate and spread among populations. Mutations start to accumulate, leading to the emergence of genetic variants. We started hearing about these genetic variants late 2020 when the UK variant B117 was described, and soon after that, some other genetic variants have been reported, such as P1 in Brazil, B1351 in South Africa, and now some other variants that have been circulating in the US, such as the New York City variant and California variants, and most recently B1617 coming out of India. And so why is this important, and why is there so much talk about these variants of concern? The reason of this is because this can impact our ability to diagnose the virus, to treat the virus, and to also be able to track the virus. And Kristen covered a little bit in terms of how this impacts our testing capabilities and diagnostic capabilities. As the virus accumulates mutations, these mutations may be present in the regions where primer binding sites are in these diagnostic tests. And this was confirmed with the B117 emergence, that some of the RT-qPCR tests that were available in the market were experiencing PCR dropouts, leading to false negative results and missing some cases of COVID. Additionally, this can impact the antigenic region that is targeted by vaccines and therapeutics. And this has been observed already with B1351 variant coming out of South Africa, in which the efficacy of neutralizing titers is reduced by 6-fold. Additionally, this can impact the viral fitness, at which could lead to higher transmission of the virus, which is again observed with B117 and other lineages that have emerged after that. They are able to transmit much easier, and they also can impact the virulence of the virus itself with some of these mutations that arise. So just to bring it a little bit into the perspective of what has been going on, in particular in the US, the CDC has noted 2 particular spike protein mutations, L452R and E484K, as variants, as mutations that are of therapeutic concern. These are present in some of these variants that I have talked about. In particular, they are in 15% national proportion of the reported genomes. In this mutation, E484K is present in B117, and also South African lineage and P1 lineage from Brazil. And L452R, it's on 7.6% national proportion and is observed in the California lineages and now the new emerging lineages from India. Additionally, just this reiterates why it's important to not only know if the virus is present in populations for diagnostic purposes, but also our ability to surveil the virus and trace it among populations. So in the US, when B117 was emerged, our sequencing surveillance efforts were well below 1%. Soon after that, we've been able to ramp up our sequencing surveillance. And as you can observe in the graph on the left side, this is data coming out of the CDC, where they are reporting the different ratios of these variants of concern among across the US. And as you can see at the very beginning, from February 2021, B117 proportion among what was being reported was very small. And as of last week, we can see now that B117 is a variant that is dominating mostly the US with about a 60% prevalence. There's also, we can see some differences in the dynamics as time keeps passing. Some of the variants, such as B1526, which emerged in New York City, it has been more predominant in the Northeast, but has been gaining a little bit of traction on Central and Western US. And then P1 also that emerged in Brazil has been gaining some traction in the US. So with this, this shows the importance of us to be able to understand what are the dynamics of the virus among populations. And as Kristin very nicely described earlier, we have two main approaches to sequence the virus. One is an Amplicon-based approach, which contains this pool of PCR primers that generate amplicons that tile across the genome, and then you prepare your libraries for NGS sequencing. Hybridization capture, which is the technology that we have validated and co-developed with Twist, it uses biotin labeled probes, and these probes are able to fish out or pull out the virus out of the background that is human background, microbial background that is present in a clinical specimen. And this allows us to amplify the signal and has been denoted by WHO now in 2020 as being a technology much more suited for detecting novel variants, but it's definitely a bit slower of a workflow. So with that, as Twist had developed already the workflow and the SARS-CoV-2 NGS panel, we partner in a matter of for months, we were able to optimize this kit for nasopharyngeal specimens, validated it on clinical specimens, and then we also built a COVID-Dx pipeline and software with an IT infrastructure that we co-submitted to the FDA. The assay was we built and launched an RUO version of this assay that was launched in November 2020. This one provides great insights into the virus and genetic variants, and this has been used now in Europe and US labs to provide and track genetic variants. Additionally, we've recently received FDA approval for our test, for our presence-absence portion of this test in March 2021. So just to give a recap of what this end-to-end solution looks like, we started and validated this assay for nasopharyngeal specimens. These nasopharyngeal specimens are collected in viral transport media, which is the transport media of choice. Once the samples are collected, they come to the lab. Samples are extracted for RNA using SIMO Direxol DNA and RNA mini prep kit. Soon after that, RNA is converted to cDNA using NEB enzymes, and this is followed by the construction of libraries using TruSeq compatible libraries from Twist, followed with the target enrichment with the SARS-CoV-2 NGS panel. Once libraries are prepared, then these libraries are loaded into an Illumina NextSeq 550 and using local run manager. After the sequencing is done, the sequencer provides you demultiplex files for each sample. Those demultiplex files, users can upload them into our cloud-based software using our Biotia portal. the software is going to take those FASTQ files and translate them into a user-friendly clinical report or a research use only optional report. And so I just want to go now a little bit in detail of the infrastructure that we have built for this particular assay. And so we have built this Biotia user portal, which is a cloud-based software that is very user-friendly. Users are able to register, both as clinicians and researchers are able to register through our portal. It is a very user-friendly. It contains a sample submission portal. This portal is HIPAA compliant with a two-step verification process. We built and connected it to a LIMS system, and it's also, for us that we're located in New York, it provides HL7 reporting to New York State. The user is able to create an order and submit an order using the serial number that is provided with the Twist SARS-CoV-2 NGS assay. That serial number is allocated depending on the type of test that the user is using, whether it's a diagnostic application or a research application. Once the user generates their order, it is submitted through the portal, and our software would analyze the samples and generate reports. The user would be able to have all of their order information and reports kept in a centralized location. When the reports become available, the user is able to look at all of the data associated with the sample that has been submitted with the submission form, and they have the ability to both download the files as a batch or as individual downloads. Additionally, we not only provide the diagnostic EUA report, we also provide the optional RUO report and FASTA files if researchers are more interested in looking more in depth into the virus. Then I'm just going to walk you through what our pipeline and outputs the user would get from using our solution. As I mentioned, we have an EUA component that is a presence-absence report. This report would generate, will detect the virus at a copy number of 800 copies per ml. This limit of detection is much more sensitive than some other platforms available in the market. Additionally, the user can receive a RUO optional report that would provide further insights on the virus and genetic variants. And this portion of the report hasn't been approved by the FDA. So in this case, what you're seeing here on the second figure is an output on our report where the user is able to get pangolin lineage of the sample that they have tested, in addition to their next clade classification and notes on how many variants that are characteristic of the particular lineage were detected within the sample. We also provide a genomic map of the specimen that was tested. So in the bottom portion, you can observe a sample that was run. The sample is analyzed at 5x depth and had a 99% coverage of the genome at that depth. In the top panel of the bottom figure, there is the genomic map. It contains the sample coverage in dark blue. In addition, we provide all of the genetic variants that are detected within the sample with the nucleotide substitution denoted in red. And in the bottom panel of that bottom figure, you can observe the percent of G-state strains that have reported that particular variant. And our most recent update was on 4/22/21. Not only we provide the genome, like the graphic representation of the genome, but we also give the user a detailed report on the genetic variants that are detected detected, giving the variant location in the genome, in which gene it is present, what nucleotide substitution was observed, and what amino acid substitution that leads to. And finally, we give a phylogenetic analysis providing information on the origin and diversity of SARS-CoV-2 based on the genetic variation. As you can observe on the left hand side, we have a phylogenetic tree. This contains all of the GSAT sequences that were deposited as of April 22, 2021. And in this case, we then give the specimen that was tested, where it falls within all of these samples that have been deposited in GSAT. Additionally, we give the frequency of the clade identified in the countries that have reported it, ordered in such a manner from the most abundant, in which country it is more abundant. Sorry. And finally, for the user, we also provide the assay performance. So basically all of these next generation sequencing metrics that the user may be interested in knowing, such as the total number of reads per sample, the percent coverage of the genome at different depth, going from 1x to 100x, and the mean target coverage. So now I'm going to switch gears to talk a bit about our clinical validation and how that looked like, the results for our clinical validation. So we tested a total of 71 positive SARS-CoV-2 specimens, and we compared those to a comparator assay of an RT-qPCR. We observe a positive percent agreement of 96.7% and a negative percent agreement of 100%. And again, as I mentioned, our limit of detection was for presence absence was 800 copies per ml, which is much more robust than some other platforms. And just as a reminder of how the virus is mutating, I previously presented a figure showing a B117, and you could observe many mutations on the coverage plot denoted in red, whereas with this sample, which was a sample collected in April 2020, had much less genetic variants present in the sample, denoting how the virus is mutating as time keeps passing by. We also wanted to gain insights into how the CT values of the samples that we validated with our assay performed on the NGS assay. So we got a total, an average of 50,000x fold enrichment of the virus among all these 71 samples that were tested. We got 93% coverage of the genome at 5x for all samples, and 48 out of 60 samples with 99% coverage of the genome at 5x. In the bottom panel, what I'm showing here is violin plots denoting the distribution of the samples, in particular looking at how the CT value impacts the percent coverage of the genome at different depths. And we were able to determine that any sample that has a CT value of 30 or below have great percent coverage of the genome, at 99% coverage of the genome, and this goes all the way up to like 100x or so. So when we did this clinical validation, we started our validation on late spring and along early summer, and we tested it on samples, nasopharyngeal swabs that were collected in New York City and Tennessee. Of these subset of samples, we were able to detect 733 mutations at 258 different mutation sites. Within the sample set, at the timeline of the pandemic, we detected 107 new mutations that were not previously reported in GSAT, and 24 of these mutations were located in the spike protein. The most frequently detected variants were C241T, which is located at the 5' UTR, and then the spike protein mutation D614G, and these two mutations emerged early in spring in the pandemic, and they have become fixed among the viruses that are circulating at the moment. So additionally, this is a heat map representing the clinical samples that were validated with the assay. And in the top panel, what you can observe is the viral genome for all of the samples. Anything that is denoted in light green is that the sequence present at that particular loci is the same sequence that is observed in the Wuhan strain or wild type strain. Those that are denoted in dark green represent nucleotide substitutions that had been reported in GSAT at the time that we did this analysis. And in purple, you can observe the new mutations that had not been reported previously. Most of the mutations were detected on the ORF1ab gene. That is no surprise, of course, because it's one of the biggest portions of the genome, but we also detected 24 in the spike protein. We detected 14 frame shifts after synonymous mutations, and 2 of these were in the spike protein. As I mentioned, we had samples from New York and Tennessee, and we were able to find a particular mutation that was only found on Tennessee samples at the time, and we have validated these results with Sanger sequencing. Additionally, as time has passed, we have tested much more specimens in our laboratory, and of these specimens, I am showing here a subset of samples that were collected in February and March of this year. And within these samples, you can observe the same genomic structure of that I was presenting in the previous figure, in which you can see that we have been able to detect variants of concern, such as B117, P1, B1351, and B1429 with our assay. And in contrast to what I was presenting previously, as time has elapsed, we have been observing more and more mutations being accumulated in the viruses that we're sequencing. And at the moment, for these particular samples, we detect an average of 34.6 mutations per sample, ranging from 27 to 41. In our clinical validation samples that were collected in April and July of last year, we had an average number of mutations of about 11 mutations, again showing how much the virus is changing as time keeps passing. And finally, I just want to go through some data that we are excited to present. We have taken this assay and have tested it in different clinical specimens, such as saliva. So as a proof of concept, we took some saliva specimens collected in New York City in January 2021. And these samples were run through our NGS workflow, and we were able to successfully detect variants in these samples with an average number of mutations of 21, ranging from 15 to 29, showing that researchers can also use this assay in different specimen types, but it may require some optimization on their end. So I hope that with what I have presented and Kristin have presented, we're able to show the power of hybrid capture in detecting novel variants, in particular because this approach allows for more mismatches due to twist technology of 100 base pair double-stranded DNA probes. This allows for a more efficient capture of the target and enables more robust variant detection as the virus continues to evolve and accumulate mutations. This is a limitation of amplicon-based approaches because the primers are much smaller, and then they're not able to tolerate as many mutations as a hybrid capture approach. This has been shown with some RT-qPCRs that targeted the spike protein that led to false negative in B117 lineage, but also it is observed in some in silico analysis that has been done by Danny and Taki and Twist Biosciences, in which he took the RTEC primer sets and looked at all of the sequences that were depositive in GSAT. And with these sequences, he was able to determine that on the primers, there's a lot of mutations that have been reported already that are falling within these primer binding sites. And what you see here on the x-axis is how many base pairs from the 3' N. On then the y-axis, you can see the number of mutations that were detected on those primers. So we're about like 5,000 mutations or so for each different base. And if we look more in detail into the variants of concern, in particular, B117 does show a lot of mutations on the RTIC primers. Again, pointing to one of the downfalls that amplicon-based technologies have in detection of novel variants. And we at our lab in Brooklyn, in order to try to understand this, we have been running some amplicon-based approaches and 2 independent nasopharyngeal samples that were collected in New York City and that were processed using an amplicon-based sequencing workflow. They were first called to be within the same lineage, which was lineage R1. And these 2 samples had a PCR dropout at the beginning of the spike protein. And so it happens when we start looking further into this, this particular dropout falls within a primer binding site of primer set 71. So this shows that as the virus continues to evolve, dropouts will increase further, and this will hinder the ability of amplicon-based approaches to detect some of these novel variants. So with that, a hybrid capture approach provides a much more uniform coverage of the genome and is able to tolerate a higher percentage of mismatches on probes, allowing for robust detection of novel variants. The end-to-end solution that we have presented reliably detects variants of concern and a wide range of genetic mutations along the SARS-CoV-2 genome. Researchers are able to adapt this workflow to process other clinical specimen types, such as saliva, and have great success in detecting SARS-CoV-2 and calling genetic variant with this solution. And finally, we are working on automation to increase throughput and streamlining the workflow for managing higher volume of samples in a 384-well format. So with that, I would like to thank our amazing Biotia team that we have worked very hard this past year, and our Twist collaborators, in particular, all of these lovely people that we have been able and have the chance to work with during this time. Our collaborators at the University of Tennessee, Robert Borstein at Lenco Laboratory for providing us with clinical samples. Agnes Berkey did a lot of the work in sequence annotation, and IAVI, SUNY Downstate, and BioBat, which are the incubators where we're part of in New York City. So with that, I will leave it there, and please feel free to reach out in any of the social media networks or personally to my email if you have any questions. Thank you.
Thank you very much for that presentation, Mara. As a reminder again to our audience, please do not forget to submit your questions to the speakers using the Q&A tab. And as we enter our Q&A session, our audience has submitted several questions, so let's get to them. And for our first question, I would like to address this to Mara first, and then perhaps after Mara has responded, Kristin could possibly weigh in as well. And our first question is, how long is this workflow from start to finish, and how does that length compare to PCR-based assays?
Kristen, do you want to take that?
It's up to you. I guess I can start. Yeah, the SARS-CoV-2 NGS workflow, this usually takes about from the start when extracting your sample and then going through library prep, hybrid capture, and so on. The wet lab portion part of it takes about 2 to 3 days. And then adding it onto an NGS platform and having that run takes, I believe that takes 18 hours. And then after that, the analysis portion. So I would say total, it takes about 4 days. Would you say, Mara, that's about?
Yeah, it's more so that amount of time. The sequencing time, it's with the NextSeq platform is 18 hours, and then once the user gets the multiplex files and load them through the portal, it is a matter of a couple of hours to get a clinical report back.
Yeah.
Thank you for that response. For our next question, I was wondering, can the hybrid capture approach compensate for potential upstream issues, for example, variations in sample processing, storage, transport, degradation, because we are dealing with clinically relevant samples which may not allow for collection uniformity. So does the hybrid capture approach offer any advantages in that regard, in compensating for that, or just being better suited to deal with that compared to other approaches?
Do you want to take that question, Mara?
Yes. So yes, like you have pointed out a very big problem that we have faced with this during the pandemic. So first of all, at the start of the pandemic, the supply chain issues were horrible. So people were starting to get creative in the types of like transport media that they could use. So we have validated our assay on different types of like viral transport media, not just one. And it definitely impacts a lot how the sample is collected, first of all, and then how it's transported into the lab. We have like the guidelines are for samples to be processed within 72 hours. If they are not processed within 72 hours, they should be frozen so that the sample doesn't degrade. And the hybrid capture, as any other assay, like it is a sensitive to mismanagement of a sample as any other assay. So I wouldn't say that, it's superior or not. The user needs to follow clinical specimen handling and, the molecular biology guidelines to ensure that their sample integrity is good. I hope that answered the question.
Yes, thank you very much. My next question is, how many samples can be run at the same time?
I believe so. Right now, the assay is meant to run 96 samples at once. So we prep 96 libraries, and then we use a multiplexing system in the hybrid capture step. And so you're making 96 libraries, and then during the hybrid capture step, you're performing 12 captures total.
Thank you very much.
And we have done with that, Nathan, we have done some proof of concept experiments. That's why I pointed out that we are looking into automation to try to scale up. So we've done some proof of concept experiments to see if we can scale up to 384 well format to process that amount of samples, and we've successfully gotten the assay to work on this. So we're talking about possibly adding that into the validation of the assay.
Thank you very much. Mara, concerning the phylogenetic tree data that you showed, I was just curious. You presented, you said that the software gave you kind of national level data correlation or data sourcing, that you could tie a specific sample to various countries of potential origin and things like that. Does the data exist on a regional or subnational level?
We don't have it as a regional or national level. It's more of a global database. This is we use GSAT, which is what most researchers have been using for depositing data. And so we are able to know where the samples are coming from, but what we give is information on at the country level.
Okay. And I think we have time for one more question, and this question is a bit more open-ended. And it's, of course, addressed to both of you. And the question is, how could this technology be used for future pandemics?
Yeah, I think this is a really interesting question, actually. I think there should be no problem. As we said, or I said in the presentation, we already had the workflow down. And so really, it was just it was making some optimization tweaks here and there throughout the protocol, but really it was just designing this panel that was specific to the virus. And so, in any future pandemics, just as long as we're able to identify the sequence of the virus that we're trying to target, there should be no problem. I think that this would be really helpful in the future for, the use in pandemics to come. I think that this is a really great piece of technology that's really going to help out some researchers. Yeah. Did you have anything to add there, Mara?
Yeah, so I do think that is this workflow is very translatable to any infectious disease. It's very powerful, in particular with viruses, due to their small genome size. So we're able to get very good target enrichment. I wouldn't foresee any issues on it being translatable to any other emerging infectious disease. And in particular, the only limitations that we could face would be the optimization based on the specimen type or any peculiarities about the particular agent that we would be testing for, but it wouldn't be much of an issue to translate it into other agents.
Thank you very much. Unfortunately, that is all the time that we have for today. If you have any further questions, please consider reaching out to the speakers directly. Their emails are shown on the screen. As a reminder, this webinar will be archived on the scientist's website, and you will receive an email notifying you when the on-demand version becomes available for viewing. I'd like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the scientist, I'd like to thank our speakers, Kristen Butcher and Mara Cuoto-Rodriguez, as well as our sponsor, Twist Bioscience. Thank you, everyone, and have a pleasant day.